Okay, so I would like to thank you for inviting me to show our work and also for joining this nice community. I'm going to show today some of the work that we are doing in collaboration with the Spanish Undiagnosed Rare Diseases program. And this program basically enrolls patients that have rare diseases and have been looking for the cause of their disease for a long time. They basically tested many things and they all came negative. So what we do is sequencing their genome through whole genome sequencing or exome sequencing. So in about 40–50% of patients, we found a mutation in a gene that has been connected to a disease that correlates with the patient phenotype, and then we can give them a diagnosis. But in a small percentage of cases, what we see is a variant on a gene that is poorly described or not correlated with a disease, but that we think that it's potentially pathogenic, but it needs to be tested to finally be able to give a diagnosis to the patient. So to do this, we develop cellular models, functional assays, and also, if needed, model organisms. So just to give you an idea of the type of patients that come to our program, most of them are children that have some sort of neural problems, from intellectual disability to behavioral issues, epilepsy, motor disorders. And that's why it's very important for us to develop good models to test neural function. And patients that got diagnosed, many of them, about 50% of them, have some issue with gene expression regulation. This will include mutations in genes involved in regulating epigenetics, transcription factors, but even genes that participate in signal transduction. Sometimes, if they are mutated, they affect the expression of many genes. So relevant to this conference, about 0.6% of the patients that join our program are diagnosed with a mutation in EHMT1. So today I'm going to present one of these cases in which we have localized a variant that is potentially pathogenic, but we need to really test. This is a child that has, we call it a CRESTA-like case. He has many symptoms that overlap with KS syndrome. This is patient number 95 in our cohort, and even his facial features resemble KS syndrome. But for this patient, we were not able to find any alteration in EHMT1 or any other gene that has been connected to this syndrome, but we were able to spot a variant in EHMT2. Dimitri already told you about EHMT2. This is also a methyltransferase that heterodimerizes with EHMT1, also very known as GNA. And what we found is missense mutations that changes alanine 1077 to serine, de novo mutation, and this variant is at a very low frequency in the population. And also, if you test like prediction softwares, it will tell you that it has a higher chance to alter the structure and maybe the function of the protein. So this alanine in green is located in the catalytic domain of EHMT2, and it's very close to other two residues that has been reported to be mutated in cancer and to confer gain of function to the protein. Actually, this area and this alanine is very much conserved between methyltransferases in humans. If we look at the structure of the protein, according to the data that has been generated, this alanine is not exactly on the very nucleus of the catalytic domain, but might be important for interaction with histone tail residues, threonine 6 and arginine 7. So we think that maybe this variant will be changing the affinity of the methyltransferase for the histone tail. For some of our patients, in addition to genome sequencing, we do RNA-seq in blood. So here we have a cohort of 40 patients in which we have profiled this gene expression. And we were interested if in our patient, there were major changes compared to other patients. We also have a patient that we took as a kind of reference or control. This is the case 120, which has been diagnosed in our program with a deletion of a single nucleotide in EHMT1, so it has a frame shift. And what we see is that there is a lot of variability in our cohort, and according to this variability, levels of EHMT1 or EHMT2 expression are not significantly different from other patients, and also the ratio between the two proteins, between the messengers, is not really much different. If you look at the principal component analysis, so these patients are the green and the blue ones, and you can see that they fall in the majority of patients. So if you check differential expressed genes, you don't see much change. So I was a bit disappointed by this data, but thank you to our collaborations, we were able to profile the methylome of this patient, the DNA methylation profile, and Dimitri already told you about it. This is an episignature. Basically, you quantify the methylation levels on an array that has more than 1,000 methylation sites, and each patient will generate a very specific profile. So in this way, you can identify up to 70 rare disorders, or even if you have many patients, you can even identify a profile that correlates with a mutation on a particular domain of a protein. So this was done in Dr. Bekin's Sadikovic laboratory. And here we see in green healthy donors that have a particular methylation profile. We see Kleefstra syndrome, which are the ones in red. And our patient is closer to Kleefstra syndrome than the healthy population, but it has its own methylation profile. Unfortunately, we don't have more patients, so we cannot establish like a cohort to elaborate as a signature. So we were kind of encouraged by this data, and we set up to generate iPS from fibroblasts from our patients. We use a Sendai-virus strategy. These are very powerful virus, and they have the advantage that they are not integrative, so they won't introduce mutations or insert in the patient's genome. And basically what you do is you infect these fibroblasts, and after 8 days, you move the cells to cell typical embryonic stem cell media, and by day 12, you start seeing colonies. So we reprogram these fibroblasts using a healthy control and also patient with the EHMT1 deletion. And what we saw is that at 11 days, we were able to spot colonies on the EHMT1 patient. Actually, this is very early. After some days, we could see the colonies also in the control. And after 19 days, it's very clear that the colonies of the patient are much bigger. So this might be correlated or in agreement with some reports that suggest that inhibiting this methyltransferase activity may improve the rate of reprogramming. However, our patient is really giving us a hard time. It took 3 reprogramming experiments to get some colonies. And what happens is that these small colonies at day 14 start appearing, but after some days, they just, if you try to pick them, they won't be able to be picked. If you leave them, they will die. So we end up having very few colonies after 32 days, really long compared to the other 2 situations. So we hope that we were now able to expand these cultures. And for the other, for the control and the KS patient, we have characterized, we have isolated more than 10 clones, but we have characterized only 2 because of obvious reasons. And you can see that the colonies have a good morphology. They are APP positive. We even check that control is the control, and the patient has this deletion, this T deletion, so we didn't mix up the samples. And for the other patient, well, we have now passage 1 colonies, so they look like this, but let's see with time if we manage to establish these cultures. So an important thing is that these IPS are free from transgene expression, and these are non-integrative viruses, but have been described to be in the cells for a long time. So you can see here that there are some clones that have passage 9, they don't have the virus anymore, but then there is a high variability because there is another clone that at passage 11 still has the viruses. So we need to give these cells several interfere with the differentiation ability. So we also check that they express pluripotent C genes by real-time PCR. We have also some immunos going on. They are very similar to human embryonic stem cells. And we are also making EVs. So this line from the KS patient is able to make EVs. So at this particular point, they don't seem very different from the control lines. And while we wait for the colonies of our patient with EHMT2 variant to grow, we set up a differentiation protocol with control IPSs obtained from healthy individuals. Actually, this protocol is modified by a colleague, Isabel Liste, and from the Lancaster protocol. And the difference is that we don't make EVs. We just add a neural induction cocktail to the cells that are growing as colonies. And after some days, you can see that they start forming these 3-dimensional structures. You can actually pick them and culture them in suspension. And finally, we keep them in a differentiation media. And after some days, we can see this structure here where more mature neurons are in the surface and immature neurons are more in the inside. So we did some preliminary analysis, and this is RNA-seq data, and it's just in bulk. So this, for each time point, contains several organoids which are pooled. And compared to fibroblasts, we can see that there is activation of very important transcription factors for neuronal specification. And we also see that compared to fibroblasts, the expression of EHMT1 and EHMT2 is it's increased. And if we look at single cell data published by others, we also found that the EHMT1 and EHMT2 expression seems to be very high in particular neuronal populations. These are early progenitors. They also express many of the genes that we see in our RNA-seq experiment. And those early populations are not anymore present in more mature organoids. So we kind of think that maybe these 2 proteins, maybe they are important for specifying these very early progenitors, but this is a hypothesis. So what we want to do when we have these organoids derived from our patients, we want to do this single-cell RNA-seq to see which specific populations seem to be affected by the variants. So at some point, we will also need to generate isogenic cell lines, corrected lines, to demonstrate that this variant is causing the phenotype. And we are also setting up an assay, in vitro assay, for checking the effects of this alanine to serine change in the EHMT2 activity. We are also profiling histone modifications in some of the patient samples. So basically, I want to thank these two ladies, Ariadna and Marina. These are master's students, and they spent the last 5 months of their lives in the cell culture room. They generated all these IPS lines. They did a great job, actually. And so these are the lines that you have seen, and now they are struggling with the other patient, but I'm sure it will be okay. I want to thank my colleague, Beatriz Martinez Delgado. Actually, the organoids, the brain organoids were obtained at her lab, and she has been very much involved in this project from the beginning. Also, our collaborators that allow us to do the profiling of the methylation in our problem patient. And we are collaborating also with researchers at the University of Sao Paulo because they may have more EHMT2 patients. And this is a call for you all. If you are in contact or you are part of diagnosis programs and you have patients with potential EHMT2 variants and you want to collaborate with me, so please give us a call, send us an email. So we will be very happy to have more patients. Try to establish a signature for DNA methylation that helps also to diagnose other patients. So please take this into account, and thank you for listening.
Thanks. Thank you so much for showing this intensive work. And this EHMT2 puzzles us already for several years. We also had many discussions about the role of EHMT2 in the pathogenesis of the syndrome and looking for individuals with variants. And yeah, then we came across your case. Are there any questions, Dimitris?
Don't give me a hard time.
Thanks. Very impressive, but I'm actually a bit off topic. So I'm just curious, how many patients do you have in the Spanish UDP? And you said that you have about 0.6% of Kleefstra syndrome in there. And I'm just curious, how come that they were not diagnosed right away, but ended up in the UDP?
We have more than 300 patients, so 300 and something. Our budget is very limited, so unfortunately, we have to select very well patients. So we try to select those that we think that they really have a genetic problem. The thing is that sometimes tests are negative and they have really mutations. So these patients that we have diagnosed with KS syndrome should have been detected before. So I think this is getting much better because now genetics units in hospitals can get more access to DNA sequencing. So I think now all these patients will be detected at that phase. So somehow there is a trend. We are moving from sequencing genome to doing more like functional genomics because a hospital calls and says, okay, I have this variant. To me, it's very suspicious. And I need you to tell me if you can develop a model or assay to confirm that. So I think it's good because this means that more and more hospitals have access to sequencing. It's more affordable. More people are gaining experience in analyzing. So short answer, they should have been detected, but they were not.